Hello everybody, today we are going to be talking to you about biochemistry. In these next few slides we're going to be understanding the structure and function of biological molecules. Sucrose is the most prevalent carbohydrate used by plants to transfer carbon. Sucrose can dissolve in water while retaining its structure. Plant cells can then export sucrose into the phloem, a unique vascular tissue that transports sugars. The sucrose is absorbed by these cells and enzymes begin to break it down into its component parts. The 6-carbon glucose and fructose molecules can be broken down into 3-carbon molecules, which are then imported into the mitochondria and processed through the citric acid cycle. This procedure decreases coenzymes, which are then employed into the oxidative phosphorylation pathway to produce ATP. Glucose can create amylose molecules, which are lengthy chains with thousands of subunits. Condensation reactions bind glucose units together, producing 14 glycosidic bonds. Because of the way the glucose units link, amylose molecules tend to form coiled springs, making it highly compact.
Lipid molecules are distinguished by the fact that they do not dissolve in water. Lipids are therefore essential for constructing structures that must keep their shape in the presence of liquid, such as cell membranes. This is also why lipids are an important component of hormones, which must pass across a liquid media. Lipids are ideal for long-term energy storage due to their strong molecular interactions. Ester bonds provide lipid molecules their insolubility. They are substances created by removing a hydrogen atom from a water molecule and combining an alcohol and an acid. A fatty acid is formed when a lengthy chain of linked carbon atoms bind to a carboxyl group, the most basic sort of lipid polymer. A carboxyl group is made up of a carbon atom that forms a double bond with a single oxygen atom and a single bond with another carbon atom. Saturated and unsaturated fats in plants and animal sources are made up of these chains. A phospholipid is formed when a triglyceride swaps one of its fatty acid chains with a phosphate group. A phosphorus atom is linked to oxygen atoms to form phosphate groups. Phospholipids have a bilayer structure with water repellent, known as hydrophobic layers, sandwiching a water permeable, also known as hydrophilic center. Cellular and intracellular membranes are made up primarily of them.
Proteins have 4 stages of structure, the first one being the protein's primary structure. This is the unique sequence of amino acids in a polypeptide chain and is the fundamental structure of the protein. Next is the protein's secondary structure, which is formed by local folding of the polypeptides in specific places. The helix and the pleated sheet structures are the most prevalent. The helix structure is present in both forms. Hydrogen bonds keep the helix of the protein's secondary structure in place. We then have the protein's tertiary structure, which is a unique 3-dimensional structure of a polypeptide. Chemical interactions on the polypeptide chain contribute to the structure, whereas the interactions among R groups are primarily responsible for a protein's complicated 3-dimensional structure. Finally, we have the quaternary structure. Unlike the previous 3 structures that only have one polypeptide chain, multiple polypeptide chains, also known as subunits, come together and give the protein its quaternary structure.
Formation and breakdown of polymers. The chemical methods by which cells generate and break down polymers are essentially the same, despite the fact that each type of polymer is made up of different types of monomer. These processes in cells are aided by enzymes, which are specialized macromolecules that speed up chemical reactions. The reaction connecting monomers amplifies a dehydration process in which 2 molecules are conveniently linked to each other with the loss of water molecule. When two monomers form a bond, each contributes a piece of water molecule produced during the reaction. One contributes a hydroxyl group, OH, while the other contributes a hydrogen atom, H. Polymers are disassembled into monomers via hydrolysis, which is the opposite of dehydration process. The process of breaking down water is known as hydrolysis. When the water molecule is added, the hydrogen from the water attaches to one monomer and the hydroxyl group attaches to the other, breaking the bond between the monomers.
Molecules must collide under ideal conditions, which enzymes can assist in creating. The glucose molecules and phosphate molecules in glucose 6-phosphate, for example, will remain linked without the presence of a suitable enzyme. The glucose and phosphate molecules, however, dissociate when the hydrolase enzyme is added.
Enzymes limit the amount of activation energy required, allowing reactions to take place at lower temperatures than they would otherwise. This accelerates the reaction time. An enzyme-substrate complex is generated when a substance attaches to the active site of an enzyme. The activation energy is reduced by the creation of the enzyme-substrate complex. This can occur in one of two ways. If two substrate molecules need to be linked, connecting to the enzymes keeps them closer together, minimizing repulsion between them and making it easier to bond. Fitting the enzyme into the active site also places a pressure on the substrate's bonds if it catalyzes a breakdown reaction. Because of this tension, the substrate molecule is more easily broken up.
Many enzymes' active sites aren't exactly the same shape as the substrate, but they do mold themselves around it when the enzyme-substrate complex forms, according to experimental findings. The active site is the right structure to catalyze the reaction only when the substrate attaches to the enzyme. As the reaction's products develop, they become less well-fitting and slip away from the active site. The enzyme reverts to its relaxed state without the substrate until the next substrate model appears.
Factors affecting enzyme activity. The enzyme and substrate no longer fit together when the active site changes shape. The enzyme becomes denatured at this moment and ceases to serve as a catalyst.
When the pH is above or below the optimal pH, the H-positive and OH-negative ions found in acids and alkalis can disrupt the ionic bonds that hold the enzyme's tertiary structure in place, causing the active site to change shape and the enzyme to be denatured.
Many hazardous compounds produce harm by inhibiting or overactivating enzymes. A competitive inhibitor like cyanide is an example. It works as a competitive inhibitor against the mitochondrial enzyme cytochrome C oxidase. This disables the electron transport chain, the final stage of cellular respiration, preventing the cell from producing ATP for energy. Tissues that rely substantially on energy, such as the CNS, brain and heart, are more vulnerable. Penicillin is an example of a non-competitive inhibitor, since it works by attaching to the bacterial enzyme. Transpeptidase is an enzyme that the bacterium utilizes to catalyze the creation of peptidoglycan crosslinks in its cell wall. It can't build new cross connections without this enzyme. Cell lysis will result as a result of perforations in the cell wall.